Hi everyone, I'm Kai Chan. I'm a visiting scholar at Harvard University and co-founder of a gene therapy startup, Ally Therapeutics. I'll be giving you a brief overview of immunology and gene therapy vectors today. Some of the concepts here might be obvious to some of you in the audience already, but our goal is to establish a common baseline knowledge and terminology prior to the workshop and discussion. Here's an outline of my presentation. I will first begin with a high-level overview of the immune system and its different arms, namely the innate immune system and the adaptive immune system. I will then move on to describing the two commonly used gene therapy vectors today, AAV and lentivirus, with a focus on the former, and describe some of the immunology known about them. Often, we don't know about the immunology of these viral vectors in the eye specifically, in which case I will describe studies outside of the eye as a proxy. Okay, to begin with, why do we have an immune system? Our immune system actually protects us. It identifies foreign microbes, mounts a response, and eliminates them. This actually protects us from the numerous viruses, bacteria, parasites, and other microorganisms that we're exposed to on a daily basis. It's actually very effective. That's why we're not catching a cold every single day of our life. But as with everything in life, you need to strike a balance. If the immune response is insufficient, you can get a persistent infection. It's chronic. It can persist for a long time. Sometimes it can even spread to areas that you don't want to, such as the brain. It's called encephalitis. Or it might allow aberrant tumor cells to proliferate. If it's too much, you might have inappropriate immune responses to things that are harmless, like allergies, or even an immune response to a self-antigen, as in the case of autoimmunity. In the case of gene therapy, the viral vector is meant to be therapeutic, but the immune system doesn't know the distinction between a therapeutic virus and a pathogenic virus. So in certain scenarios, viral vectors can also cause too much immune response. This is a one-figure overview of how our immune system works. Broadly speaking, it is split into innate immunity and adaptive immunity. So initially, when a virus infects us, some innate immune cells will traffic to the site of infection where there is viral replication, and they try to triage that infection. This happens in the acute phase. We're talking about minutes, hours, and the first days of infection. And what these immune cells are really trying to do is to slow down the replication and spread of the microbe to mark the site of that infection. And they also chop up the microbe into many pieces. Subsequently, there is a link. The innate immune cells present those chopped up microbes to B cells and T cells, and also stimulate them to proliferate and improve their specificity for the microbes. So days to weeks later, you now get antibodies and effector T cells that are highly, highly trained against the invading microbe. How does the innate immune system work? So like I said, they're the first responders, and they do so by recognizing some kind of conserved signal, commonly a conserved signal from microbe, commonly known as pathogen-associated molecular pattern, or PAMS. And this is basically something that cannot be mutated away by microbes. This is something that will always be different between human and microbes. Here are two very, very classic examples. The first one is lipopolysaccharide, LPS, also known as endotoxin. This is a major component of the cell wall of gram-negative bacteria. Immune cells use a receptor known as toll-like receptor 4, TLR4, to detect it. And consequently, LPS is highly, highly inflammatory. The other example here is nucleic acids in the wrong cellular compartment. So for example, in human cells, we keep our DNA in the nucleus and in the mitochondria. So if there's an incoming packet of DNA in the endosome, that is a sign of microbial invasion, and that can be detected by a receptor known as toll-like receptor 9, TLR9. These innate immune cells, they sense the microbe. They can eat up the bacteria or the infected cells. They can secrete cytokines and chemokines, activate and bring in more immune cells. And they can also trigger cells in that tissue to stop transcription and go into an antiviral state. So this slows down the infection and the replication of the microbe while the innate immune cells kickstart adaptive immunity. Okay, how does adaptive immunity work? So this comes on later, but they are highly, highly specific and highly, highly potent. This is an educated immune response such that it specifically recognizes the invading microbe. For example, you might have an antibody that recognizes a specific H5N1 strain of influenza, but not that of a different influenza strain. They do so by using processes known as VDJ, recombination, hypersomatic mutation, etc. Essentially, they mutate and create a protein receptor that is very, very high in affinity for that exact microbe. This adaptive immune system is further split into two arms, the humoral arm and the cellular arm. The humoral arm is when a B cell produces and secretes antibodies. These antibodies can bind and neutralize viruses, don't allow them to enter a cell, or they can mark the microbe for destruction. Cellular is when a cytotoxic T cell recognizes an infected cells. Those infected cells can present viral peptides on MHC class 1 on the surface. And now the cytotoxic T cells are licensed to directly kill those infected cells. This is advantageous because you limit the ability of infected cells to spread virus. But this is also potentially dangerous and damaging, especially for neurons because they don't regenerate. Finally, adaptive immunity has something known as memory, while innate immunity doesn't. So if your immune system has seen a virus, it can often remember it for a long time because the B cells and T cells after the infection persist as memory cells. So they're ready to expand and spring into action again if they see the microbe in future. Okay, so that was the immunology primer. I'll now switch gears to talk about gene therapy vectors and what is known about their immune responses. I want to first start off with some quick history. The gene therapy field started out with two popular vectors in the past initially, but there were safety concerns. The first one, adenovirus, it has a huge coding capacity, but it's also highly inflammatory. And when it was intravenously infused into a teenager called Jesse Gelsinger to treat a disease called OTC deficiency, it resulted in a lot of inflammation and death in several days. There's a different vector, a gamma retroviral-based vector. This is used to transduce hematopoietic stem cells. It integrates into the genome. It was infused, retroviral transduced cells were infused into kids with a disease known as X-linked SCID. That's an immunodeficiency in a French trial. And the kids essentially had their immune systems restored, but multiple kids developed leukemia eventually, and one died. So if you fast forward to today, the picture looks very different. We now have highly efficient vectors that have a much more favorable safety profile. With AAV, it is mostly used for in vivo gene therapy. With lentivirus, a different type of retrovirus, it's mostly used for ex vivo gene therapy. Correspondingly, the two of them have lower immunogenicity and lower oncogenesis concerns. AAV has seen some very high-profile successes in recent years with 3 approvals. These are targeting indications that affect the muscle, affect the retina, as well as neuromuscular compartments. And AAV gene therapies appear to be very promising. There are many, many in development, and the FDA estimates that there will be 10 to 20 cell and gene therapy approvals per year by 2025. However, everything has its limits, and certainly that is the case for AAV as well. Just in the past year, there have been several treatment-related serious adverse events observed for AAV, particularly when it is administered at a high dose intravenously, systemically. For example, on the left, Solid and Pfizer, they both had patients experience complement activation. That's a kind of innate immune response. And it was accompanied by acute kidney failure. This was for their Duchenne muscular dystrophy trials. Solid has been placed on clinical hold twice now. And you can also see all of the other SAEs I've curated from all of the various high-dose systemic AAV trials. In recent months, the safety concerns continue to increase.
Audentes has reported 2 deaths, and actually just this month, a 3rd death. This is 3 deaths out of 17 boys for the high-dose cohort, not 4, apologies for the mistake on the slide. And these are the first ever treatment-related deaths for AAV.
So if you ask me, we're still learning about the safety and immunogenicity issues with AAV, especially since many of these trials are for rare genetic diseases. They're small trials in which we're starting to test and treat more patients. The risk-benefit consideration is really important here. If it's a severe disease with no alternatives, there might be more tolerance for safety and immunogenicity signals. If it's a non-life-threatening disease or those with treatment alternatives, that calculus can change. Obviously, ocular AAV gene therapy doesn't have life-threatening toxicities, but we hope that with this workshop, we can openly discuss what kind of immunogenicity signals we're seeing. Here's a one-figure summary of immune responses to the AAV vector. In terms of innate immunity, 3 sensors are implicated: toll-like receptor 9, toll-like receptor 2, and MDA5. The main one appears to be toll-like receptor 9 for AAV, as that has been studied and verified by multiple labs. For adaptive immunity, much of the work has been focused around antibody responses to the capsid, the protein shell of the vector, as well as cytotoxic T-cell responses to the capsid. Let's now go through each arm. Starting with innate immunity and Toll9, here's a model for how Toll9 interacts with AAV. Toll9 is expressed in the endosomes of immune cells, such as plasmacytoid dendritic cells. When AAV is administered in vivo, it can be endocytosed by these cells, and the DNA genome of some viral vectors are presumably exposed at some point, and Toll9 detects that incoming DNA, a sign of microbial invasion. Toll9 then activates and triggers the induction of innate immune responses, such as inflammatory cytokines and interferons. And it is these signals that eventually lead to the formation of anti-capsid T cells. Let's look at some data. Here are some mouse studies. Immune cells from Toll9 knockout mice don't mount an innate immune response to AAV vectors in vitro, unlike wild-type cells. If you administer AAV intramuscularly to mice, wild-type mice develop a T-cell response against capsid, while the knockout mice have a greatly reduced T-cell response. With both intravenous and intramuscular delivery, note that all of this work was done in mice, Toll9 knockout mice appear to express higher transgene expression, and it also seems to persist better than wild-type mice. However, these knockout mice eventually develop antibodies against the capsid, similar to wild-type mice, as shown here. So this suggests that other innate immune factors must exist and contribute to antibody formation. There are some limitations with all of these studies, which is that while multiple groups have shown the importance of Toll9 for AAV, they were mostly testing mice in the context of intravenous and intramuscular routes of administration. So it is not so straightforward to extrapolate to subretinal and intravitreal AAV in humans. 2 other innate immune sensors are implicated for AAV, but the evidence is sparse. On the left is toll-like receptor 2. This typically detects bacterial byproducts. There is a single publication by a German group showing that anti-capsid, AV2 and AV8 here, can trigger toll-like receptor 2 mediated inflammation. It is currently unclear what toll-like receptor 2 is recognizing on the AAV capsid and how it does it. Now, on the right is MDA5. This usually detects long double-stranded RNA in the cytoplasm. This is a hallmark of replicating RNA viruses, since humans only make single-stranded RNA. A group at UNC showed that expression of MDA5, gene expression, is upregulated after AAV transduction. And they postulated that the viral ITRs, the two viral ITRs, are directing expression of both a positive sense RNA transcript and a negative sense RNA transcript, and that leads to double-stranded RNA. The evidence around MDA5 is more circumstantial. I think toll-like receptor 2 is more likely to contribute to the AAV immune response. So far yet, there isn't quite clear evidence in the literature, though, on how it impacts the adaptive immune response to AAV in vivo. Okay, next up, I'm going to transition to adaptive immunity for AAV. Let's start with anti-capsid antibodies. That's typically measured with a neutralizing antibody assay, a NAB assay. This is a major concern for systemic AAV administration because even low amounts of NAB will block viral transduction. But with subretinal AAV, shown here as a clinical study, it has been shown that one can inject one eye subretinally, and then a few years later inject the contralateral eye without issues. So this suggests that at least for subretinal applications, anti-capsid antibodies is not really a key concern for subretinal AAV. For intravitreal AAV, that's not as immune privileged as subretinal. So neutralizing antibodies in the vitreous might be a concern. For anti-capsid T-cell responses, most of our understanding actually come from intravenous AAV applications that have targeted the liver. This is a very classic example. On the left, a patient receiving an intravenous injection of AAV at first shows an increase in the transgene expression. This is the red line. In this case, it's a clotting factor known as factor 9. Weeks later, you get an asymptomatic increase in transaminases, ALT and AST, which is indicative of liver inflammation. And concurrent with this inflammation, you start to see that the transgene expression wanes and is finally eliminated. This is thought to be due to a cytotoxic T-cell response against the capsid. This can be measured by taking PBMCs from the patients and performing an ELISpot assay. And this also led to trials now testing and adopting prophylactic corticosteroid protocols. The key concern here for intravenous applications is durability of transgene expression. However, with intraocular AAV applications, it seems that usually the T-cell response against capsid is low, sometimes not detectable, at least when PBMCs are assayed. It also seems to be quite transient in nature. Okay, two more quick concepts on AAV immune responses. The immunity to AAV can be split into pre-existing and induced immunity. For pre-existing, that's because wild-type AAV, it's naturally out there. It can naturally infect us, so many of us have antibodies already that can sometimes be used as an exclusion criterion in certain gene therapy trials. With an induced immune response, that's a de novo immune response. I already talked about how one could potentially see antibody and T cells directed against the capsid. As we test more ocular gene therapy applications, we should also consider an immune response against the transgene product, which is the encoded protein. This risk could be higher when the protein is not a self protein or a wild-type protein. For example, it could be of microbial origin, it could be some kind of unnatural fusion protein, or it could be a humanized monoclonal antibody that originally came from mice. Also, there are some patients that don't express the wild-type gene at all. They're known as null patients or CRIM-negative patients. So they don't even have tolerance to the wild-type protein, even if it was completely wild-type. The next point I'd like to make is about systemic immune responses versus local immune responses. As you can imagine, most assays for immunogenicity are utilizing the blood. And that's just because it's convenient. It's easy to access this repeatedly. That also means that we're really measuring immune responses outside of the eye, which is different than local inflammation. It is not completely clear what the relationship is between that peripheral immune response and the immune response inside the eye. So when you have good results in the peripheral assays, it doesn't necessarily guarantee that you won't have an immune reaction in the eye. On the right is a striking example of why the location of sampling really matters. This is a non-human primate study done by Jim Wilson's lab. They treated some monkeys with intravenous AAV8, and when they took out T cells from the blood, they found very little immune response to the transgene. If they took out T cells from the liver or the spleen, you see a very, very different story. The immune response was dramatically higher. When it comes to local inflammation, intraocular inflammation in the eye, the clinically apparent inflammation can be detected through slit lamp examinations. You can look at aqueous cells, vitreous cells, or you could use some kind of imaging technique to look at the retina. Still, nothing beats analyzing the tissue itself. So for something that is not clinically apparent, this would not be possible in a clinical setting. Histology of the tissues might elucidate some pathology as well as some immune infiltration, and measuring immune factors in the humors might also tell us more. This is my last slide, and it's on immune responses to lentiviral vectors. Lentiviruses are actually RNA viruses, but upon entering a cell, they reverse transcribe the RNA genome into DNA, which then integrate into the human genome. This means that it has a dual RNA and DNA lifestyle, meaning that it also activates both RNA and DNA sensors, in this case, Toll7 for RNA and Toll9 and cGAS for DNA. In terms of adaptive immune responses, similarly, one could get antibodies and cytotoxic T cells against the envelope protein, and one might also be concerned about adaptive immune responses to the transgene product. This is the end of my introductory presentation to immunology and gene therapy vectors. Thank you for your attention.